All right, so now we know how IGF-1 or insulin can activate mTOR. So the question then is, does resistance exercise, what does it do to IGF-1? And this is work out of Dan West, who was a student of Stu Phillips at the time. And what he did is he did resistance exercise in individuals, or he kept them in a control state. So these are the same individuals at two different visits. And what he did was he measured IGF-1, circulating IGF-1 after they did resistance exercise, really heavy resistance exercise of the legs. So between rest and zero time exercise, they did heavy resistance exercise on the legs. And what you can see is within 15 minutes, there's a nice 50% increase in IGF-1 levels. Okay, it doesn't last a hugely long time, but again, this is heavy leg exercise, but not a lot of forced repetitions. But it just means that if you do a short bout of resistance exercise, you increase circulating IGF-1 levels. So, maybe now IGF-1 is driving this response to resistance exercise. If we look at a much more strenuous exercise pattern, these are animals that have been hypophysectomized. So that means they've taken out the hypothalamus, and so there's no growth hormone here. So the idea is this because of an initial growth hormone response? So is it a circulating effect where the IGF-1 is coming because you've increased stress, you've got more growth hormone, and that's giving you IGF-1? Or is it something specific to the muscle? So what they're looking at here is IGF-1 and IGF-2 mRNA within the muscle at 2, 4, and 8 days of this overload hypertrophy in rats. So this is an extreme model of muscle growth where you can almost double the size of a muscle in 14 days in a rat by taking out other muscles that normally are doing the work. But what you can see is that within 2 days, there's a huge increase in IGF-1, and that continues out to this 8-day time point. So now this is where we start, or people started to develop this idea that what happens is that the heavy load causes an increase in stress locally within the muscle that increases local production of IGF-1, and that local production of IGF-1 is going to feed back and give us muscle hypertrophy. So to test this, a group out of Texas, what they did is they just took an animal, and all they did is they added IGF-1 so that the muscle now produces IGF-1 constitutively. So at a very high level, you increase the amount of IGF-1 being produced by the muscle. All right, and so all they did is they had control animals, or they overexpressed this little piece of DNA that had IGF-1 in it, and what they saw was quite dramatic. And you can see it here. You went from this muscle to this muscle, and just looking at that, you would say that's got a lot more steak in it. And if you actually cut it down the middle and you turn it on its side and you stain the different muscle fibers, this is the control gluteus muscle. So that's the butt muscle of the animal. And what you can see is you had these nice fibers, and they're good sized and they're really cool looking. But then if you look at the IGF-1 overexpressing muscles, that's a huge increase. That's a huge increase in muscle fiber size. That's over a 2-fold increase. We're looking at a 2.5 to 3-fold increase in muscle fiber size. So you can see that this one here versus this one here, you've got just a huge increase in area, and that's muscle hypertrophy. The area of the muscle fiber is a lot bigger. So this was really exciting at the time. You see the date, 1995. If you go back into the track and field world, a little bit after that, what happens is this incredible athlete comes along by the name of Marion Jones, and Marion Jones was one of the greatest Olympic athletes of all time. She was a great, great sprinter and a great long jumper, and she was this incredible athlete, such an incredible athlete that she was also a basketball player, and she went as a freshman to the University of North Carolina and turned them from an average basketball team to the national championships. The national champions. So she's an incredible athlete. But then these things start circulating, and the way that bro science works is, IGF-1 causes muscle hypertrophy. Yes, that's good. You read the abstract of the paper, and you see that IGF-1 causes muscle hypertrophy. So what do people do? They start injecting themselves with IGF-1. There's a problem with that. When you inject yourself with IGF-1, that's a protein. It circulates, and as we saw in the West paper, the protein can go up. In the West paper, it went up 50%, but then it was already back down to normal 15 minutes later. So you had this huge increase in protein that goes away within 15 minutes. So if I inject myself with IGF-1, 15 minutes later, 30 minutes later to an hour later, it's all gone. They didn't read the paper. If you read the paper, here's the important part. It's highlighted in red down here. That when they made these mice, the muscle had 47-fold overexpression of IGF-1. So that was 47 times more IGF-1. When we did the resistance exercise, we got 50%. That's 0.5-fold. So we're looking at two orders of magnitude difference in the amount of IGF-1. And the other thing that's really important is that IGF-1 is 24 hours a day, every day from the point that you made your first muscle cell. So you have this on all the time at a hugely high level. You get muscle hypertrophy. That's different than when you inject yourself with IGF-1. Even if you have a long-lasting IGF-1, it's not going to have this same effect. So this fit the hypothesis, though everybody's all excited. Okay, resistance exercise, increase IGF-1. IGF-1, yes, this came before 1999 when we knew that mTOR drove muscle hypertrophy. But when that data started coming out, now it's IGF-1 is driving mTOR and mTOR is driving a muscle to get bigger. So let's actually test this. And this is work that was done at UC Davis by Espen Spangenberg, who's now left UC Davis, unfortunately, but he still continues to do good science. And what he did is he took what's called the muscle knock-in receptor. These are animals that had a knock-in mutation to their insulin or IGF-1 receptor. And the IGF-1 receptor, therefore, couldn't become activated by insulin or IGF-1. And so what they did is they looked at wild type muscle mass, and that's right here, versus the knock-in mutation muscle mass. Look at that. There's actually no real difference in the muscle mass of these animals. Then what they did is they did that functional overload where they cut out a muscle and they let the muscle grow, and you can see that it gets almost 2 times bigger after 7 days, and it gets to 2 times bigger by 35 days. If you look at the animals that didn't have IGF-1 signaling at all in them, their muscles cannot turn on the IGF-1 signaling pathway. They had the exact same increase in muscle hypertrophy that the wild type animals had. So that suggested that IGF-1 isn't needed for the muscles to grow in response to load. So load-induced muscle hypertrophy, maybe it doesn't need this. What one of my PhD students, Lee Hamilton, did is remember the first step in the insulin IGF-1 pathway. The insulin IGF-1 pathway, the first step is recruitment of IRS-1, and then P85 comes to IRS-1. So when insulin binds to the receptor, you can see that the P85 associated with IRS-1, IRS-2, or both of them, the phosphotyrosine here, it goes way up with insulin. It goes up 100 to 250 times. So 250%, sorry, 1 to 2 fold. Okay, so when you inject insulin, you get this nice increase in IRS-1 associated with P85. That's what I already showed you. That's how insulin activates mTOR. What happens after resistance exercise? So what we did here is did a bout of resistance exercise, took 20 minutes, and collected the muscle either immediately after or 30 minutes, 3 hours, 6 hours, 18, 48 hours later. And what you can see is that insulin caused this increase in association of P85 and IRS-1. Resistance exercise actually decreased it. And there was a 50% decrease in association of IRS-1 or IRS-2 with P85. And so what that says is that in a completely normal animal, not like the transgenic animals that Espen's group used, what happens is you're activating mTOR, because when we look 3 hours after exercise, mTOR phosphorylation of S6 kinase is really, really high. But it's not happening by IRS-1 associating with P85 and getting PIP2 to PIP3. It's happening in a different way. And so this is the first evidence in wild type animals that resistance exercise is activating mTOR in a different way than IGF-1. So really, in summary, IGF-1 is produced by muscle cells after resistance exercise. It might not actually function in the muscle. It might actually have a different function. If you increase IGF-1 in muscle 47-fold, 24 hours a day, 7 days a week, for the whole life of an individual, muscle will grow significantly bigger, up to 3-fold bigger. However, muscle hypertrophy occurs in mice lacking a functional IGF-1 receptor, and loading-induced growth happens in an IGF-1 independent manner.